will present Ayari Fuentes Hernández. She got an undergraduate degree in physics from UNAM University, Mexico, and a PhD in mathematical biology from Imperial College in London. She did a postdoc at the Department of Biosciences in University of Exeter, where she started to work in antibiotic resistance. Since 2014, she's assistant professor in system and synthetic biology at the Center for Genomic Sciences, UNAM University in Mexico. And her lab combines mathematical modeling with experimental evolution to study the effect of temporal and spatial structure in the ecological and evolutionary dynamics of bacterial populations. Her work focuses on the evolution of antibiotic resistance and their effect in bacterial communities. And so I leave you with Ayari, please, you can start your presentation.
Well, thank you. First of all, thank you very much for the invitation of all the organizers to put together this really nice conference. And then what I'm going to talk to you about is a bit of the work in my lab that we have been doing about single-cell microfluidics and image bioinformatics to study plasmid dynamics. So first, I want you to introduce you why I'm doing this. And the point is, I think we all know that antibiotic resistance is a very important health problem. And this is because although antibiotics are really useful, and actually they change our life like radically. Antibiotic resistant bacteria are on the rise, and we have a void of antibiotics of new antibiotics introducing in the clinic in the last decades. So it's a really big problem because today actually we haven't found any antibiotic that is actually proved to all bacteria, and we have found bacteria that are resistant to all the antibiotics that we have. So in that sense, a lot of labs actually I have been trying to understand even in lab setups how emerging and why. So although plasmid has been really important in this. Almost all outbreaks in hospital setups are actually driven by plasmids, and antibiotic resistant genes can often carry by them. So a plasmid is a small DNA molecule that can actually replicate, and sometimes they can introduce in the chromosome. They can carry beneficial genes for the host, like nitrogen fixation, and the case that we are interested in is drop resistant genes. And they can also transmit between hosts, even in different species. And that is why plasmids are really successful actually to transmit antibiotic resistant genes and via horizontal gene transfer. And of course, plasmid is is associated with some fitness cost. So, carrying plasmid is costly in the sense of the bacteria is not going to be able to grow as well as without the plasmid. So, of course, they can be lost through segregation, and I'm going to get to there a little bit further in the talk. And now the experimental system that we developed to research this is going to be a multi-copy plasmid, which is going to be in average 19 copies per cell. It's going to be non-conjugative, which means that it cannot transfer horizontally, and is going to stay within the host. And it's going to carry one antibiotic resistant genes within the BLATM1, which is going to confer resistance to ampicillin in particular. And we are going to mark it with a fluorescence marker that is going to be a GFP, so it's going to glow in green. And we are going to put this system of multi-copy plasmid in an Escherichia coli K12 MG1655. So with that in mind, we are the first thing that we did is we did a little bit of experimental evolution, which is like we grow our Escherichia coli in a plate, then we transfer just one clone of that into fresh media, homogeneous and liquid fresh media, and then we are going to transfer every day into one part of that population into fresh media with everything new. we are going to start transferring it. And then at the end of 16 days, we are going to see what happened to the population and what happened if the population is the same as the one that we started with. This is really nice because what we can do is we can actually put in a big fridge, like minus 80 centigrade, and that means that we will have samples, frozen samples of everything. So we can go back and we can, we can actually see what happened within every day. The other thing that we do is, what we want to see is how this bacteria with this plasmid can adapt to antibiotics, actually. So we take this bacteria and we are going to introduce it to one antibiotic. So we are going to do what we call a ramp, which is every day with the media, we introduce a bit of antibiotic, like the first day is going to be 0.25, the second day is 0.5, and then 1. We are going to measure it in MIC, and MIC is the minimal inhibitory concentration, which is the last concentration at which we found actually growth in our samples. So what we see, with what's really, really interesting, is that we start with a population with this 20, with this system, with this Escherichia coli, with this plasmid that confers resistance to ampicillin, and we introduce another antibiotic, which is ceftazidime, and we know that ceftazidime is the same class of ampicillin. There are beta-lactams, and we also know that the TEM1 that we introduce is just one mutation from the resistant gene that confers resistance to this other antibiotic, this other beta-lactam, which is ceftazidime. So we introduced ceftazidime in a ramp, and after 16 days, we found that actually this population is really, really resistant, like 8,000 times more resistant than the initial population. So, well, we found that at the group in a group with which we are collaborating with in Madrid, from Álvaro and Millán. What they did was that this really nice experiment that where they compare this system with the plasmid with another 2 strains. One with the wild type without any resistant gene, which we are going to call in here MG, and we are also going to compare it with the MG blood TEM1, which is a strain, a Escherichia coli strain, which is going to have one copy of the plasmid, of the gene, but in the chromosome. So we are going to compare the one that has the gene, the resistant gene in the chromosome, the one that is carrying the plasmid, and the wild type. What they did, they do the same experiment of 16 days doing a ramp-on antibiotic of ceftazidime, which we are going to call it CAS. And this was really, really interesting because the first 2 populations, the MG and the MG blood-TEM, the one that has the gene in the chromosome, are going to start collapsing actually in the 6th day when the MIC is around 8. They behave really, really, really similar. And which means that actually the one copy in the chromosome is not conferring any resistance to this antibiotic. But in contrast, when we have the MG PBGT, which is the one that has the plasmid, after the 16 days is going to be very, very resistant. It's going to be 8,196 times more resistant than the one at the beginning. But also is really interesting because the number of population actually surviving decreased a lot, which means that maybe the plasmid is, well, the plasmid is going to be very costly, although they've conferred, they conferred a lot of resistance. So then we wanted to know what happened here within the plasmid dynamics, and then we did a mathematical modeling. So what I'm showing here is in the top graph, we are showing the number of copies of the plasmid, and in the bottom one, we are showing the plasmid type. So we have that at the beginning for 4 days, we are going to have the plasmid that we put on in around 20 copies, as I told you, and then in the 4th day, we are going to have a mutation in the plasmid that is going to confer the resistance to this new antibiotic. Remember that TEM1, it was just resistant to ampicillin, not to ceftazidime. And then for some point from 4 to 8 days, what we have is a combination of the mutant plasmid and the wild type plasmid. And then the mutant plasmid, which is better for the environment that they are in, they are going to fixate in the population. And then interestingly, what we found is that after the new plasmid is going to fixate, it's going to start increasing in copies. And we are going to have around 320 copies of this plasmid. So maybe that is why they found really small numbers of surviving population, although they are really, really resistant. So this is the experimental data. And so we know, well, we can say that multi-copy plasmid can act as enhancers of rapid gene amplification. And then, interestingly, from 4 to 8 days, we have that this period of time that we have the coexistence between the parental and the mutant allele, and we found this really interesting. So we wanted to know what is the consequence of having this this part of heterozygosity. And then, well, this is the dynamics within one cell. So we have a cell that have the plasmid, the ancestral one, the wild type, which is all the ones in black. And then we have the one, the mutated one, which is the yellow one. And then when they start dividing, we can lose it by segregation, of course, but particularly if we have the right environment, this is going to start fixating in the environment until all the population is going to be like that. And sorry. So when we want to study this, what we did now is to develop another experimental system. And now we have the same plasmid with the blood TEM1, which is marked with the GFP. And then we are going to have the same plasmid, but now we are going to introduce this new mutant gene, which is going to be the blood TEM12. And this is going to be marked with the DS red. And then we are going to have, well, these two is going to be in two different cells, of course, but then we are going to have also cells that we are going to put these two plasmids in the cell. So they have the same origin of replication. So in principle, they are kind of the same cells, the same plasmid. So then they can actually coexist really well in the same cell. So we did this. So now we have three different populations, the one that we are going to call R12, which is the one that has the blood TEM12, the G1, which is the one that we already have, that has the blood TEM1, is the one that has the combination of both of them. And then we wanted to see if there is any difference in resistance. So in here we are seeing checkerboards where in the x-axis, what we have is the increase of ampicillin. In the y-axis, we have the increase of Cas. So the population of G1, which is the one that just have the blood TEM1, is just resistant to ampicillin. The population of the R12 is just resistant a bit of ampicillin, but is also very resistant to Cas. And then we are going to have a mixed population, which is going to have a population of half G1 and half of R12, just together in the same amount. And it had multi-resistant to ampicillin and to Cas. And then the HT population, and the HT population are cells that have both plasmid within the cell. So if you can see here, actually, we cannot differentiate a lot the mix to the HT. Like they kind of have the same profile of resistance. So why is evolutionary stable, or why they appear to be these heterozygous populations? So what we wanted to know is what is the evolutionary benefit of presenting this intracellular genetic diversity. And now what we did was a bit of experimental evolution again. And then we have these two populations put in together, and we transfer these populations within these 16 days, as before, doing a ramp of Cas. And what we found is that the population of G1, which at the beginning was in the 90%, it was the 99% of the population, decreased and collapses very quickly. But the one that is fixated is the population of R12. So this is not surprise because the environment is Cas and R12 are resistant to Cas. So what happened when we have a population, another experiment, which we have 98% of the population which is G1 and a 2% of the population that is HT, the heterozygous population. And what we have is really interesting because at the beginning, the population of G1 actually collapses at the same level as the one in the top. And at the end of the experiment, the R12 is going to fixate in the population exactly the same as before. But now we have this HT population, the heterozygous population, that is going to increase in frequency and then is going to collapse. So by some, exactly by some amount of time in the population, we are going to have coexistence of the G1 and the R12 in the population, both within a cell. So this is again, this was just a model, a mathematical model, sorry. And this is all the experimental data and which fits quite nicely. And then what we are saying here is that multi-copy plasmid is promoting transient coexistence between parental and mutant alleles. So what I'm saying is that these windows we found really, really interesting. So now we wanted to find what happened with this window of transient coexistence, both within a cell. So by that, we wanted to quantify this plasmid-mediated heterozygosity. And at the beginning, what we did is just have a sample of a population, and with flow cytometry, we can actually see, we can differentiate the three different populations, the R12, the HT, and the G1. Just with the differences of GFP intensity and DS-RED intensity. So what we wanted to do is now to have a representation of just two parameters of plasmid. So we wanted to have the plasmid frequency and the plasmid copy number. So we did a change of just coordinates to angular coordinates, and we have this really nice representation. And now, in this representation, how we can see how are seen these three different populations. So the R12 is going to see just in the left of this arc. The HT is going to be a broader, well, almost everything is going to be in the middle, but it's going to be really dispersed in all the graph. And the G1 is going to be all to the right. But then this is, as I told you, this is just a sample of the population at the end of the population. But we wanted to see what is the intracellular plasmid dynamics. So in order to see this, we use a single cell microfluidic now. And we use it is called a dual input model machine. So this is really nice. This is a really small chip for PDMS, which is just a silicon. And what we did is you introduce bacteria here, and you trap just in the top of this channel, you trap what we call a mother, and then we just start putting fresh media. It's a continuous media device, and then they start dividing, and you see through a microscope, to a fluorescent microscope, so we can actually have images in GFP and also DS-RED. Once we have this, we have three different images: the DIC, which is normal light. This is GFP, which is the green one, and DS-RED, which is going to be the red one. And then we do well, we first have to do image analysis in order to see what are the frequencies within one cell of the plasmid. So do a segmentation image like here, and then we use we have to do a mask of those cells. We have to see which one is a cell, and we have in order to track it. For that, we use a deep cell algorithm, which is an open source algorithm of computational learning, and then we do by hand a corrected mask. So even the deep cell is actually a really good algorithm, but it has these kind of things that we have to clean by hand. So we correct the mask, and then we can start the cell tracking. And by the cell tracking, I mean we identify one cell, and then we can follow all the daughters of the cell, and we can have the cell division, we can measure times, and of course what we really wanted to know is the relative intensity of each cell. And then, well, this is just the image, that the process image, and as we can see, if everything goes everything looks really green, where the things not the population, the cell is going to have more green plasmid, which are the ones that have the blood temp 1, and if there is more red, the cells are going to be more are going to have more plasmids of the blood temp 12. And now, if we measure it through time, which is part of the interesting thing about microfluidics, that we can have time lapses, like really long ones. What we see here is that each of these lines is one mother cell, which measure, and all of these points are when they're divided. So we measure the relative intensity within division, and to see how is the difference within segregation of the plasmid. As you can see here, sometimes are a bit more red or a bit more green, but this is quite random in the sense that it's not changing a lot. And this is just one cell, and all the other ones are all the other ones that we measure. So it's actually not a preferable pathway through G1 or through R12. So we can measure this and with the relative intensity, and this is really normally distributed. So what we are saying now is the segregational drift is a noise-driven process. So it doesn't, it's just a random process. But this is without selection. So what we wanted to do is, well, this plasmid actually confers resistance to different things. So are they going to behave differently with selection? So in order to measure selection, we are now going to use a microchemostat, which is another microfluidic device. Now, instead of these really tiny lines, we have really big chambers. So we can trap around 800 cells, more or less. And the really nice thing about this is that we can actually change the environment in a really precise way. So in here we can put some antibiotic and remove the antibiotic, and we can measure that in real time. So we did that, and with the heterozygous population. And now in the top one, we are going to see one of these chambers, and we are going to put an ampicillin in there, and in the bottom one, we are going to put Cas. So we are going here.
Ayari, sorry, yes. You have 5 minutes, sorry.
Okay, no, that's fine. And then, well, we see this, and the important thing here is that we see a lot of changes of colors, and the important thing here is at the end of the experiment, almost everything is there. So what we are seeing here, and what we can measure, is that in the top one, when we are introducing ampicillin, there is a shift to GFP, and in the bottom one, when we are introducing Cas, there is a shift to the other one, to DS-RAD. So now, if we have those two, what is going to happen when we have fluctuating environments? So now what we are going to do is to shift between Cas and AM in the same chip. And then, and this is the video that I'm showing. And the important thing here is that even though we are using the same amount of antibiotic that we were using before, we have the population alive at the end of the experiment, and they have actually, this is 18 hours, and we have population there. So what we are seeing here is that fluctuating selection stabilizes intracellular genetic diversity. And we wanted to check that, but now we wanted to check it in a population level. So now we are going to use the kind of same experiments that I presented before, and we are going to have the mixed population, but now we are going to introduce each day a different antibiotic, CAS or ANT, and we are going to compare it with just the HT population. So we have a model that's saying that you introduce in the top one, when you have the mixed population, you introduce one of the antibiotics, and the other population is going to decrease a lot. So when you introduce the AMP again, all the population is almost dead. So the population is going to collapse eventually. But when you have the HT, as we see, actually the both of the genes are there, so you can rescue the population. So they become more resistant actually to fluctuating environments. So this is all the experimental data, and where we have the 4 populations: the U1, the R12, the mix, and the HT. And the ones that I want you to look at are the dark ones, which has the one that we are changing environments. And in those ones, the HT is much more, we have more surviving replicates. So actually the intracellular genetic diversity promotes survival of bacterial populations. So just in quick summary, so what we are saying is plasmid can act as platform for gene amplification and accelerate the rate of adaptation. That segregational drift is a stochastic process resulting from randomly segregating and replicating plasmids. That selection modulates allele frequency distribution in bacterial populations. And finally, we are saying that multi-copy plasmid increase intracellular genetic diversity, increasing survival in fluctuating environments. So it's very good to be heterozygous if you are going to have fluctuating environments. So that's it. Thank you very much. And this is the people that we are collaborating with in this project. So it's Álvaro San Millán and from the CMB. And almost all the work that I presented is done by him, Charlie Hernández. He's a PhD student, and also in close collaboration with Rafael Peña-Miller, who is also a collaborator in the Center for Genomic Sciences. So thank you very much.
Thank you, Shari, for your very nice talk. And I've learned a lot of bacteria. I didn't know. It was very interesting. And I am really surprised to see deep learning here, because my question is if the deep cell algorithm you mentioned is a deep learning algorithm that works on images.
Yes, and it's a really, really good one. Yes.
Yes, and I have 2 further questions. Why you've chosen, if it is kind of a state of the art or it's something very new but that works, and how you train it? You train it on a population in one experiment, and then you test it for another experiment with another community, or how is that with this deep learning?
Yes, we actually try to do a lot of, for this tracking stuff, we try to do a lot of things, and nothing works a lot. So it was really time consuming. And then they published this, I don't know, 5 or maybe 8 years ago. And it just worked like really, really well. And you training with the, you have to take a video of the same thing actually that you want to do, and you can train it with that. So at the beginning, it's just really, well, you have to train it by hand, but then it works really, really well.
Okay, but you use one section of this video for training, and then you use another part to discover. Yes. Okay.
Yeah.
Excellent. Very interesting. Yeah, I couldn't imagine this application of deep learning, and it's really interesting.
It's really good, and actually it changes a lot, like the time when we want to measure these kind of things. So it's really cool.
Great, thank you. We have some congrats from public, and we have Evelyn, who says really great. Can you mention again the name of the deep learning algorithm, please?
DeepCell.
DeepCell, okay.
Yes.
And it's new, you said 8 years ago?
Yeah, something like that. something like that, yeah.
Okay, great. Well, thank you very much, Ayari, for this very interesting talk. Thank you, really.